Hello to our friends joining us via recording. We are starting lesson number 9 today about the muscular system. To help us get ready to talk about lesson number 9, we did an application question. So our application question, we're trying to figure out how rescue inhalers work for people who have asthma. So as we start addressing this question, our goal is to figure out which kind of muscle tissue is affected by albuterol, which is one of the medications in a rescue inhaler. I want to figure out what it causes to happen in that type of muscle tissue. So let's go up to the top with the information we have about asthma and see what we can figure out here. So when a person is having an asthma attack, what happens is their bronchial tubes get smaller. So they start constricting, their size goes down, and that allows less air to enter the lungs. Hey, for my friends who are here in class, let's make a guess. You do or do not control the size of your bronchial tubes. Do you decide how big your bronchial tubes are? Yeah, we are correctly identifying in the chat. You do not make that decision. You're not deciding, or your child or your friend is not deciding to randomly shrink the size of the bronchial tubes. If you don't decide when this kind of muscle contracts, that means that one of our 3 types of muscle tissue here cannot be the right answer. Which kind of muscle tissue do you decide when it contracts? Which kind is consciously controlled, as we call it? Yeah, skeletal muscle is the type that is consciously controlled. So when we think about an asthma attack, when we're trying to figure out which kind of tissue is being affected in an asthma attack, the first thing we can think about is whether or not you consciously decide to have an asthma attack. And we don't, because we don't consciously decide to have an asthma attack. We know that skeletal muscle is not what's being affected here. We were talking about, when we were working on this question, how during an asthma attack, our bronchial tubes shrink, or the amount of space in the airways. Here, let's do a little bit art up here. We have a normal sized airway. Here's a bronchial tube. I'm looking at a cross section of it. When a person is having an asthma attack, that size can get much smaller. We go from being very large to being very small. So my goal with using a rescue inhaler is to go from this small size back to my large size. Now, our type of muscle tissue that up to this point we haven't yet defined, but our type of muscle tissue we're talking about lives inside the walls. Apologize for my ugly drawing here. It lives inside the walls of these bronchial tubes. When that type of muscle tissue is relaxed, there's a lot of space. When I contract it, it squeezes down smaller. So when we're talking about using a rescue inhaler, are we going to want our muscle tissue to contract or relax when we use that inhaler? What do we want to go on with those muscles? Yeah, we want those muscles to relax. We want those muscles to relax because when my muscle tissue is contracted, when it's squeezing together, it is preventing, or it's kind of squishing itself down. It's not allowing those cells to have any space between them. So when they relax, when they spread back out, there's more space in the middle. So we know that we want our muscle tissue to relax. We haven't yet decided if it's smooth muscle or cardiac muscle. One of these types of tissue I only find in one place in the body. Which type of muscle tissue, smooth or cardiac, is only in one place? Yeah, cardiac's only in one place, right? So cardiac muscle is only found where? Yeah, the only place I find cardiac muscle is inside the wall of the heart. Absolutely. So when we're talking about the bronchial tubes, which is the small tubes inside the lung, that the lung is not inside the wall of the heart. It can't be cardiac muscle. It tells me that the type of muscle we're talking about is smooth muscle. So the way that a rescue inhaler works is it sends a chemical message to the smooth muscle that lives inside your airways. It tells that smooth muscle, hey, let's relax, chill out. We need more space for air to come into the lungs. We had a great question during our conversation here. When you look at your tissue location and function guide for smooth muscle, it says that smooth muscle is found inside hollow organs. When you think about what hollow organs are just an organ inside your body that has space inside of it. So the word hollow means empty, or means that there's a space inside something. So a hollow organ is any organ with space inside, we'll say, with space inside. So one of those organs that we talked about, I think I've told you guys this. Which one, do we happen to know which organ system is my favorite organ system? Have we talked about that? Yeah, digestive system is my favorite system. Digestive system is completely filled with smooth muscle. So some of my hollow organs are things like the intestines, where there is space for food inside of. We churn it around, we absorb the nutrients out of it. What is the hollow organ that has blood inside of it? Not the heart. What's the hollow organ with smooth muscle that has blood inside of it? Any ideas? I promise it's not as hard of a question as it sounds. I did a really bad job explaining it. Yeah, we're just talking about blood vessels here. That was a very bad explanation. I apologize. Blood vessels are considered another hollow organ, quote, quote, because they have space inside of them for blood. Another hollow organ, like we're talking about in our example here, are the lungs. Inside those bronchial tubes, we have space for air. So when you're thinking about an organ that has space inside of it for something to be in there, whether it's air, whether it's blood, whether it's food, some kind of liquid, the muscle tissue that's found inside of it is probably going to be smooth muscle tissue. So I know you all love my application questions, right? But now we understand asthma a little bit better, right? So hopefully that helps us out. And you know I do like to do some of these application type stuff on exams, right? So walk through it like we did here. We figured out what we do and don't know and used what we do know to work our way through it. Any questions about this before we move on? Yeah, Nicole said that I love questions about verbs, right? So, yep, you got to read those questions slowly. I do like to change words on you. Make sure you're paying attention. We kind of just dove right in. You all want to send me emojis? How are we doing on this Thursday? I'm going to find an emoji for today too. Here's my emoji. Not enough of this yet. Nicole's sweating. Yeah. Oh, Jesse's doing good. Awesome. Yeah, lab this week is a big one. The good news is next week is a little bit better, so that's good. But this week's lab is definitely a rough one. That's for sure. Okay, well, I've got a couple of us that are inquisitive, a couple of us that are ready to learn. So let's do a little bit of learning. The good news is too, someone mentioned that we do have a lot of review here at the beginning, which is excellent. So we're going to tie in the anatomy from lab into some new physiology here from lecture. So this particular lesson, we are learning about muscle tissue in depth. When we talk about muscle tissue, here's something I want you to underline, highlight, star. Anytime you see the beginning part sarco, anytime we see sarco, so I'm going to underline it in a couple of places here. Sarco always means muscle. So sarco means muscle. You, as you're working on this assignment, I ask you to compare some muscle words to regular cell words. So the one that we talked about in lab for sure was called the Sarcoplasmic Reticulum. The Sarcoplasmic Reticulum. Who can remember from lab what we said that the Sarcoplasmic Reticulum is? What's it like in a normal cell? Yeah, so the Sarcoplasmic Reticulum sounds a whole lot like Endoplasmic Reticulum. We just changed the beginning part there to say sarco. So the Sarcoplasmic Reticulum is the endoplasmic reticulum, specifically the smooth endoplasmic reticulum, but the endoplasmic reticulum of a muscle cell. Jesse mentioned the membrane. We do actually have a special muscle word for the membrane too. On a muscle cell, we call that the sarcolemma, plasma membrane. So the sarcolemma, this ending part here, lemma, means membrane. The sarcolemma is the plasma membrane of a muscle cell. Because anytime I see sarco, I'm thinking muscle cell. In lab, we talked about that thing called the sarcomere. The sarcomere. This is the part of a muscle cell, the part that actually contracts, the sarcomere. I don't have a regular cell word for something like the sarcomere, but I'm pointing that one out to say, here's that beginning part, sarco. So remember from lesson number 8, anytime we saw the beginning part chondro, who remembers what chondro meant? Anybody remember chondro? Yep, so chondro was cartilage. Okay. Osteo, what was osteo? Yep, osteo is bone. So we're adding to our list here. Anytime you see chondro, we're thinking cartilage. Anytime we see osteo, we're thinking bone. Anytime we see sarco, we're thinking muscle. The other thing that we see in muscle cells, the other tip-off word for us, if you will, is myo. So if you see myo, or there's some words from lab, things like mesium. So let me add that to our list here. Myo and mesium. Words like that, or parts of a word like that, also mean muscle tissue. So you can see that in the little thing I'm putting a box around right here, myofibrils. Myofibrils are the special protein fibers inside muscle cells. So fibril means protein fiber, myo means muscle. So myofibrils are these groups of muscles that are inside my muscle cells, or these groups of proteins, excuse me, that are inside my muscle cells. These myofibrils, you can see their little stripes, right? These myofibrils are where we're actually going to find those sarcomeres. So the parts of a muscle cell that actually contract, like that picture you saw in lab, all of those are inside these myofibrils that we see here. By the way, just for a frame of reference, in case these pictures are a little bit confusing for you, or a little bit weird, this is an entire muscle cell. I actually have a special name for muscle cells, by the way. Does anyone happen to know from your reviewing? What's the special name for muscle cells? What do I call those? Yeah, so we need to know that the word muscle fiber is the fancy muscle word for a cell, muscle cell. So you're going to hear me during lesson number 9, or you're going to read in the guided lesson for number 9 about muscle fibers. That's the special name for a muscle cell. Just like we had osteocytes that were bone cells, Just like we had chondrocytes that were cartilage cells, muscle fibers are muscle cells. So we can kind of make a list for ourselves, and I actually have you do this in your lesson outline. We've got our muscle fiber, that is the muscle cell. The plasma membrane of that muscle fiber is called the sarcolemma. Oh, here, let me, we don't have it labeled on here, but there's a special name for the soupy stuff that's inside a muscle cell. Does anyone happen to know either in a muscle cell or a regular cell what we call that? Yeah, so we got our muscle cell stuff coming out. The name of the soupy stuff inside a muscle cell is the sarcoplasm. The sarcoplasm, move that up. The sarcoplasm is the cytoplasm of a muscle fiber. The sarcolemma is the plasma membrane of a muscle fiber. The Sarcoplasmic Reticulum is the endoplasmic reticulum of a muscle fiber. So we need to memorize our words so we can go back and forth between regular cell and muscle cell. That's going to help us as we're thinking about or using our muscle terminology. So Jacqueline asked in the chat if the sarcoplasmic reticulum helps us to build proteins. Here's where it would have been better for me to be more specific, so I'll add it there. The sarcoplasmic reticulum actually is technically the smooth endoplasmic reticulum. Did the smooth endoplasmic reticulum build proteins? Does anyone remember? Yeah, so the smooth endoplasmic reticulum doesn't build proteins. The rough endoplasmic reticulum does, but the smooth endoplasmic reticulum doesn't. So when we think about the function of the sarcoplasmic reticulum, it's not a protein builder. In particular, the big thing that the sarcoplasmic reticulum does for us that's going to be really important is that it stores calcium. Stores calcium. Because making the process of muscle contraction happen requires that I release calcium at the right time. So the sarcoplasmic reticulum is the place inside a muscle fiber where calcium is found. When I want my muscle fiber to contract, I spit out that calcium from the sarcoplasmic reticulum, let it go into the cell so that it can go down by all of these myofibrils and allow those sarcomeres inside of them to contract. So I don't have, to my knowledge, I don't have a special name for the rough ER in a muscle cell. It's just that smooth ER that I have a special name for, and I call it the sarcoplasmic reticulum. Yeah, Jesse mentioned in the chat that calcium is used a lot. Yeah, that's why we talked about how your body has to balance it last unit. One of the reasons I have to make sure that it's balanced at the right level is because of this, because I use it for muscle contraction. Another reason I have to make sure it's balanced is, as we'll see in the next lesson in number 10, I have to have it to let my neurons talk to each other. So yeah, calcium is really important, more than just making your bones strong. Calcium is important for muscle contraction, and it's important for neurons talking to each other. So yeah, very important for us to have our calcium. The one other thing I will mention as we are looking inside our muscle fiber here, inside our muscle cell, notice that what we're going to spend most of our time in lesson number 9 talking about is the type of muscle tissue that has more than one nucleus. Which type of muscle tissue had more than one nucleus? Does anyone remember? Yeah, so the type of muscle tissue with more than one nucleus, remember it was the type of muscle tissue I told you kind of looked like ants on a log. It's skeletal muscle tissue. So do make sure that you remember what all the types of muscle tissue look like. Do make sure we remember what the jobs are of each of those types of muscle tissue. But the primary one we're going to focus on, the way that it works anyway, we're going to focus on the way that skeletal muscle tissue works. So more than one nucleus inside these big, long, cylindrical cells. We mentioned that earlier when we were chatting. Cylindrical means you look like a cylinder or look like a tube. Lots of nuclei in these types of cells. The other thing that we have a lot of in these cells are mitochondria. Why would I need a lot of mitochondria inside this type of cells? What do mitochondria do for a cell? Yeah, absolutely. Multiple of us are chiming in. It's all about ATP, right? It's all about energy. So the process of muscle contraction requires a lot of energy. We will probably talk about today how I use energy to make muscle contraction happen. So I'm going to have lots of mitochondria inside my muscle fiber as well to make sure my muscle fiber has the energy that it needs to do contraction. How do we feel about our muscle fiber words right now? Thumbs up, or what questions do we have at the moment? Yeah, so Jessie asked a lab question. The word that she asked about was the epimesium. So let me add that word to our slide here so we can talk about it together. The epimesium. When you see this mesium part right here, mentioned that's another muscle word. So whenever we're talking about something in muscle, mesium actually technically means connective tissue. So when we talk about any kind of mesium, because there's actually multiple types, there's the endomysium and the epimesium. There's even one called the perimesium. It's always connective tissue. Do we remember, I'll put this to the class, what the beginning part epi meant? So my hint for us would be maybe, where is the epidermis compared to the dermis or the hypodermis? Where's the epidermis? Yeah, so epi is the one that means above or on top. Epi, I'm going to put above. That's the easiest way for me to remember it. The epidermis is the layer that's above the dermis. Yeah, superficial. There's our technical directional term, right? So epi means superficial. The epimesium is the part of, it's the connective tissue that wraps around an entire muscle. So all of your skeletal muscles are made out of a bunch of different cells that all have to work together at the same time for you to do a movement. The epimesium is the connective tissue that I find all the way on the outside of a muscle, keeping that whole muscle together. So when you look at a picture of a muscle man and you see the shape of the pectoralis major muscle, the fact that you see that shape is because all of those muscle cells are grouped together by epimesium. Or when we look at the legs and we see the shape of the rectus femoris muscle, you've got epimesium holding the entire muscle, rectus femoris, together. So epimesium, I'll type it here for us, surrounds the entire muscle. The other one I think that we had to know for lab was the endomysium. Ooh, I spelled that wrong. Try again. Endomysium. Endo means on the inside. So the endomysium is the special kind of connective tissue around single cells. So technically, when we look at our muscle fiber here, our muscle cell here, there would actually be some connective tissue on the outside of it as well. That connective tissue is called the endomysium. So mesium, connective tissue. Epi versus endo. Just tells me, are we on the very outside? Are we completely superficial? Or are we inside really small, surrounding each of my individual muscle cells? I believe that's just lab terminology. I don't think that I asked you about that in lecture, but that's good good review for us, because that is something we will have to know for the lab final exam. So good question. All right. Nothing new on this picture except to point out, here's that word we talked about before. What was the sarcoplasm the same thing as? Remind me. The sarcoplasm in a muscle fiber is the same as a normal cell's, the normal cell's cytoplasm. Exactly. So just mentioning, because it was labeled on this picture, that the sarcoplasm is that cytoplasm of a muscle fiber. Okay, so let's talk about, here, let me bounce back and show you what we're going to focus on. We are going to talk about the process of muscle contraction. Muscle contraction happens inside these protein tubes called myofibrils. So the myofibrils, myo means muscle, fibril means proteins. Muscle cells are filled with all of these big, long protein tubes. Protein tubes that are going to get smaller or get bigger, depending on whether we're contracting or relaxing. Inside these protein tubes, let me draw us a little box right here. Inside these protein tubes, you can see what we talked about in lab last week. When we talked about the sarcomere, we talked about how the sarcomere went from one Z disc to another Z disc. We talked about how there was an M line in the middle of that sarcomere. So we are pulling together that lab stuff from last week, looking at it inside muscle tissue now here in lecture. So each of these long myofibril tubes that you see here are made of lots and lots of sarcomeres. And those sarcomeres will do the process of muscle contraction to allow you to move a part of your body. So big picture, when we're thinking from big scale to small scale, we start off with a muscle fiber, a muscle cell that's really long. Inside that muscle fiber are these protein tubes called myofibrils. When I contract my sarcomere, when I bring my Z discs closer together, that's going to make these protein tubes shrink smaller, which makes my muscle fiber shrink smaller too. That brings my bones closer together, for example. Let's do a lab review question really fast. When I bring bones closer together, what did I call that kind of movement? Exactly. Yep. When I move bones closer together, or the way that the lab packet defined it, right, was when I make the angle between them smaller. Same thing. That's the process of flexion. So think about, keep the big picture in the back of your mind as we start talking about small scale, how the process of contraction works. Because what we're doing is we're bringing bones closer together. And the way I do that is by shrinking the protein tubes that are inside the cells that make up these big muscles. Here's some lab review, and I know we were bemoaning at the beginning of class here. My colors, unfortunately, are opposite in lecture than they were on that lab model. I have no idea why the makers of the lab model did their colors backwards, because like every anatomy book on earth is going to have them color coded like this. So you can be angry at the lab model for mixing us up here. Inside our sarcomere, our thing that does contraction, we have two main proteins. The main proteins are grouped together either as the thick filament or the thin filament. So when we talk about the thick filament, it gets its name because it's literally physically thick. If you look back at the lab model picture, this was the one that was blue and really wide. But really the best way to identify the thick filament besides the fact that it's thick, is these places right here, these little golf club heads that you see. So these are the parts of the protein myosin that make up the thick filament. That's an underline highlight star idea that the thick filament of a protein, or excuse me, of a muscle cell is the myosin protein. I build these really thick, funky looking filaments here by wrapping a whole bunch of myosins around each other. So you can see all of these individual myosin proteins that have been curled up in a long string. We end up having all of these little places where the myosin heads are sticking off of. Myosin heads are the places on myosin where it can interact with actin. Actin, if you remember from lab, is the main protein that makes up the thin filament. So we kind of see these little blueberry looking proteins. These are actin when it's by itself, and then we string it all together in these lines that we see here. That is what actin looks like inside a muscle fiber. Actin has specific places, you can see these little divots in actin here, specific places where it likes to attach to myosin. So we have places on myosin that are great for attaching to actin, places on actin that are just waiting to attach to myosin. The thing is though, and we mentioned this when we were talking about lab, we are not always interacting myosin and actin with each other. We are not always doing the process of muscle contraction. To help us make sure that we're not always doing the process of muscle contraction, we use proteins like tropomyosin and troponin to help us do that. So these are what we call regulatory proteins, tropomyosin and troponin. My contractile proteins, or my proteins that do muscle contraction, that's actin and myosin. In the chat, help me remember, what is the function of tropomyosin? What did we say in lab that tropomyosin did? What was its job? Yeah, I'm making you type long. I'm sorry. Yeah, so Gloria's absolutely right. She's mentioning that the job of tropomyosin, and you can use its name, that's how I like to keep things straight here. Tropomyosin, its job is to cover the myosin binding sites on actin. So tropomyosin, we're playing a trick. That word trope is a trick. So basically tropomyosin means I'm tricking myosin by blocking the place that it would normally attach to actin. If I can't attach to actin at these myosin binding sites, I can't attach at all. So if tropomyosin is in place, myosin can't attach to actin. No muscle contraction happens. But if you remember from lab, when we talked about tropomyosin, I said that it was kind of like a wet spaghetti noodle. So we've got this wet spaghetti noodle that is wrapping around my string of actin here, my blueberry string, right? I've got this wet spaghetti noodle that is not always going to stay in place. It's going to need some help if we're going to block those myosin binding sites. What was the name of the protein that helps tropomyosin stay in place? It kind of acts like a push pin, if you will, keep tropomyosin in place. Yeah, the one that does that is troponin. So troponin is my push pin. It pushes on this spaghetti noodle, says you are not moving. We are going to keep those myosin binding sites blocked. Troponin is great at doing its job, at holding tropomyosin in place, and tropomyosin is great at doing its job, blocking myosin from attaching, as long as I don't release any calcium into my cell. Does anyone happen to remember when I release calcium into my muscle fiber, Which of these proteins does calcium interact with? Who does calcium attach to? Does anyone know? Yeah, so calcium attaches to my push pin, to troponin. If calcium attaches to the push pin, if it attaches to troponin, think about the push pin just popping out of place. If I pop my push pin out of place, my wet spaghetti noodle, that's tropomyosin, that's going to pop out of place too. And suddenly I now have an open myosin binding site for myosin to come along and attach. And when myosin attaches, we get the process of muscle contraction. So what do I want to make sure you guys know with these pictures? First thing I want to make sure you know is which proteins live in the thick filament and which proteins live in the thin filament. That's the first thing I want you to know. And the second thing I want you to know is what the jobs of each of these proteins are. So we want to make a list for ourselves in our notes. The job of tropomyosin, the job of troponin, the job of actin, and the job of myosin. Make sure we know all of those things. Questions? Thoughts? Emojis? That's what I get when I search for muscle. So Nicole asked a good question that I'm going to put to the class. Let's talk this one out together. How might we describe the job of actin? Any ideas? What do you guys think about what we might say actin does? So I wouldn't necessarily say it relaxes the muscle. It is part of what I use to build my muscles. There's a couple of things that I would say when I'm talking about the job of actin. The first thing I would say that the job of actin is it makes up the thin filament. It builds the thin filament. Most of what the thin filament is actin. So job number one, I'm going to say, not in yellow because you won't be able to see that. Actin, I would say job number one is making up the thin filament, so building the thin filament. Yeah, and then the other thing I would say, kind of going with what Christina mentioned, actin is considered a contractile protein. So actin is directly involved in the process of muscle contraction. So I'm just going to make a note at the top too that actin we would call a contractile protein. So it's involved in contraction. Too far over there. So actin is a contractile protein. It makes muscle contractions happen. Those are kind of the two things I would say. So if I were to ask you, there's another protein that would have a very similar description. If I traded out this word thin filament for thick filament, which protein would we say makes up the thick filament and is a contractile protein? Yeah, exactly. Myosin, we could say the same kinds of things. It makes up the thick filament and it's a contractile protein. What's the name of those regulatory proteins again? Regulatory proteins, as in the ones that are not contractile proteins. That's my hint for you. Yep. So tropomyosin, the one that blocks myosin from attaching, and troponin, my push pin. Yeah, so the question in the chat is my contractile proteins are correct. It's both actin and myosin are considered contractile proteins. My regulatory proteins are troponin and tropomyosin. Troponin and tropomyosin, they don't make contraction happen. They just help us know when it's going to happen. My contractile proteins make contraction happen. When myosin attaches to and pulls on actin, then contraction happens. My regulatory proteins just decide whether or not myosin and actin get to interact with each other. Let's talk about how the process of muscle contraction works. I believe this is split up over a couple of pages. What pages in your outline do you have these pictures here? Okay, so it starts on page 6 and bleeds over to page 7, we're saying. Okay, what we are talking about here is the actual process of muscle contraction. The generic name for the process of muscle contraction is called the cross-bridge cycle. Cross-bridge cycle. So this is a note for you to make for yourself. When we talk about the cross-bridge cycle, in easy words, when we're trying to describe what the cross-bridge cycle is, it's literally the way we make movement. The way we make movement. That's the simplest way to think about the cross-bridge cycle. When movement happens in your body, my contractile proteins have to slide past each other. They have to move past each other. So the way that I make movement happen is by doing several specific things interacting actin and myosin with each other. So 4 steps to this process: formation, the power stroke, detachment, and cocking the myosin head. Let's go through and talk about how this cycle works. For me to make movement, the first thing I have to do is I have to do a step called cross-bridge formation. In cross-bridge formation, it sounds all technical and fancy. Literally all that happens in this stage is I form a cross-bridge. Now, we haven't defined what a cross-bridge is. So let me, if I can type this morning, what a cross-bridge is. All it is where myosin attaches to actin. So myosin attached to actin. That's what a cross-bridge is. If myosin is going to pull actin and make actin move, the first thing I have to do before myosin can attach to actin, or the first thing I can do before they can slide past each other, is I've got to attach them to each other. So step 1 of the cross-bridge cycle of the way that I'm going to make movement is by starting with attaching myosin, my thick filament protein, to actin, my major thin filament protein. Step 1, myosin and actin attach to each other. Step 2 is where we actually have movement. So when you're trying to figure out what happens in each of these steps, the step called the power stroke or the working stroke, this is where things actually move. So this is the step where actin actually moves. Power stroke. So what's making your muscle get shorter, allowing you to do a bicep curl? The step that actually does the movement is the power stroke. Here's what happens in this power stroke stage. So way back here in step 1, when myosin attached to actin, myosin is thinking, sweet, this is awesome. I love being attached to actin. At the same time though, myosin says, you know, when I'm attached to actin, I don't need to hold on to these other chemical compounds. So we see some other chemical compounds back here. Myosin says, I don't really need this stuff. This stuff is junk. I don't need it anymore. So in the power stroke step, myosin spits out its junk. When it spits that stuff out, that actually changes its shape. So remember, in anatomy, we always said that structure and function are related. When I take these things out of the structure of my myosin protein, suddenly its shape changes. It goes from myosin being standing upright to myosin bending over. When myosin bends over, actin has to move because myosin and actin are attached to each other. Step 1, myosin attaches to actin. It's still holding all of its junk on its back. Step 2, myosin spits out that junk, and that changes its shape, causing it to pull actin along with it. Now, at the end of the power stroke, myosin has a moment of clarity and is like, oh, I'm attached to actin, I'm bent over, I'm stuck, I can't move anymore, nothing else is happening. Well, along comes a molecule of ATP, and ATP attaches to myosin. When ATP attaches to myosin, myosin's like, pshaw, I don't need actin anymore, I'm done. It lets go of actin, and now it has a shiny new thing to play with. It's got ATP, it's attached to ATP, so it's happy. Hey, remind me in the chat, what is ATP? Why is ATP important? Yeah, ATP is an energy source, absolutely. So up here, myosin has no energy. It's stuck attached to actin. At the end of the power stroke, it can't do anything. It's stuck. It can't move. If a molecule of ATP comes along and attaches to myosin, that allows it to detach or no longer be holding on to actin. It's not attached anymore. The cross bridge is gone. Cross bridge detachment is when I detach or snip apart myosin and actin. But the last stage of the cross bridge cycle is myosin saying, hey, this energy, this toy that I have here, ATP, is super great. I'm going to split it in half to allow me to change my shape. So the last stage of the cross bridge cycle is called cocking the myosin head. It's taking the myosin head from being bent over, where it's inactive, into its upright position. Because when myosin is back in the upright position, it can then go back and attach to actin again. Now, Christina asked an important question. Christina asked if ATP is formed with ADP and this PI thing that we see here. So short answer is yes. See this right here? It says ATP hydrolysis. Hydrolysis is a fancy word for I split something up using a molecule of water. So hydro means water, lysis means breaking. ATP hydrolysis is when I chop up a molecule of ATP and I use its energy to do something for me. So in the cross-bridge detachment phase, ATP comes and attaches to them by putting something, oh, somebody's got a mic on. By putting this ATP on myosin's back, myosin lets go of actin. But if myosin can chop up this ATP, turn it into ADP and inorganic phosphate, that's going to allow it to stand back up. That's going to put it in the cocked position so that as long as actin is close and it's available, myosin could attach again. Let's do some chemistry here. We all love chemistry, right? Okay, ATP. The name ATP means adenosine triphosphate. ATP, adenosine triphosphate. Anyone want to take a guess at what ADP means? What's that D? You still got the adenosine, still got the phosphate. Any guesses on D? Yeah, Christina's right. D, adenosine diphosphate. ADP is adenosine diphosphate. Di means 2, tri means 3. So here's what ATP actually is. I'm going to move over on my board here. ATP, we've got this root thing called an adenosine. It's a chemical compound called adenosine. And then I've got phosphates attached to it. I'm going to have to make these closer. Okay. PO4, that's what the chemical formula is for phosphate, by the way. PO4, PO4. Okay, so the way that this works is in the molecule of ATP, we've got adenosine, and then we've got, let me move them. I should probably just draw instead of circles. Okay, adenosine triphosphate, ATP. We have my adenosine root or chemical that's attached to 1, 2, and 3 phosphate groups. Whenever we talk about the energy that a molecule has, the energy of ATP, where that energy is actually found is inside these chemical bonds that attach these phosphate groups to our adenosine molecule. So the adenosine molecule has 2 bonds that store energy, although in reality, really the only one that has enough energy to matter is this bond right here. So this chemical bond out here that attaches my 3rd phosphate, my triphosphate, to the rest of this parts of the molecule, this is where all the energy's at. So if my myosin protein can go through and just slice this bond, it gets to keep the energy that was stored inside here. It uses that energy to go from being bent over into the back into the upright position. But when I chop this bond, this phosphate group isn't attached to these guys anymore. So now what I've got going on is I've got a molecule of adenosine that's attached to 2 phosphates, and I've got 1 phosphate that's floating around by itself. This is what is labeled on your chart there as ADP, and then they call it PI, inorganic phosphate. When they aren't connected to each other, when I've chopped that up, now this doesn't have energy anymore. ADP, not really energetic, not helpful for me. Phosphate, definitely not helpful for me. But that energy that used to be here, attaching the ADP to that last phosphate, I use that energy to cock my myosin head to make it stand back up. So ATP hydrolysis is when I steal the energy that's stored inside ATP. When I steal that energy, I got to chop it up. That's where I make that junk we were talking about. That junk that once I attach to actin, I don't want to hold on to anymore. I don't care about it. I don't need it. I'm going to spit it out, and that's going to allow me to bend over and move my actin protein. So let me see my chat here. So your question is close. Jacqueline is basically asking about the relationship between ADP and ATP. So ATP is made by our mitochondria, right? That's where I make ATP. I spit that out, my myosin chops it up. Our cells can recycle ADP, but the caveat is they've got to send that ADP back to the mitochondria to attach it to a new phosphate. So it's not going to be the same ADP and phosphate that are connected to each other that make up this ATP that we see here. It's probably going to be a different one, but our cell will do recycling like that. Absolutely. It'll go and add another bond back inside here to connect together. We're doing recycling all over the place in your cell. And one of the things that we do recycle is our ADP and our phosphate, connect them back together, store some energy in that bond, and that would make the ATP. So they're definitely related to each other. It may not be the exact same one that goes through this cycle, but they're definitely related. Yeah, so Christina's question asks, so is that why we find so many mitochondria in the muscle? So we can trade out that ADP for ATP. Exactly right. Yes, 100%. To make muscle contraction happen, you've got to have ATP. So I need to have mitochondria to make that happen. So great correlation. Remember we were looking at our muscle cells and we saw that they had a ton of mitochondria? Here's why. Because I literally physically need ATP to attach to myosin to allow the process of muscle contraction to happen. What other questions do we have? I'm getting the crickets. Shoot me an emoji. Shoot me an emoji with your weekend plans, if you have any. I'm trying to think if I have any weekend plans. Let's see if I can find an emoji. Here's my weekend plans. I'm gonna be grading. Great fun. Oh, Nicole, are you going to the farm? Trying to interpret the gardening. Got it. Okay. Yep, I can definitely see that. Christina will be studying. Yeah, pulling weeds, right? Maybe for the last time. It's supposed to cool off. Although weeds are resilient, right? They like to grow no matter the weather. Gloria is either going hiking or shoe shopping. Hiking, okay. I was like, yes, to which one? That's fun. How many of us have had a chance? Let me look at the notes here. How many of us have had a chance to look at rigor mortis? Have any of us had a chance to look at rigor mortis? Okay, so maybe just one person. Okay, so the good news about that then is we're going to figure this out together. Okay, so let me clear off my slide here. So Gloria, you get to listen more than type in now since you already studied rigor mortis a little bit. For my friends who haven't studied rigor mortis yet, do we have any idea what rigor mortis is? Have we heard of that terminology before? Rigor mortis. Rigor mortis. Let me type it. Yeah, so rigor mortis. Rigor means that things are stiff. Mortis is referring to death. So rigor mortis, when muscles get stiff after you die. Muscles get stiff after you die. So if you ever watch any of those cop shows, right? They always like to talk about rigor mortis, right? To figure out the time of death. Rigor mortis and body temperature are the things they always talk about. So let's talk about rigor mortis. Rigor mortis, the reason your muscles get stiff after you die, has to do with this process here of the cross-bridge cycle. Okay, let's think for a moment. When you die, there's something that your body stops making. Any thoughts about what your body stops making when you die? I mean, there's a lot of things, right? There's something really important. So calcium is something that comes in. Yeah, so we do stop making everything eventually. Yep. One of the things that's our big problem though, that we stop making pretty quickly after we die, or put it this way, pretty quickly when we stop breathing, we need oxygen to make any measurable level of ATP. So ATP, the best way for us to make it is to use our mitochondria. Our mitochondria need oxygen to be built, or to work, excuse me. So to build ATP in the body, we need oxygen. You die, you stop breathing. We can't make any more ATP. So when we die, we don't make any more ATP. When we die, no more ATP made. Remind me in the chat, and you can use the picture to help you out here. What is the role of ATP in muscle contraction? What specifically is ATP doing in muscle contraction? It does help us with movement, but how specifically? Yeah, so Nicole's mentioning the specific step in the cross-bridge cycle that I use ATP is this one right here. The first place we see ATP coming into the cross-bridge cycle is a stage called cross-bridge detachment. The job of ATP is to be that shiny thing that attaches to myosin's back and convinces myosin, okay, you can actually let go of actin. Like, it's going to be okay, I promise. ATP comes in and helps myosin detach from actin. Okay, so let's go back to rigor mortis. When I die, I don't make any more ATP. If I'm not making any more ATP, what is not going to happen in my cell? If I don't have any ATP, what is not going to happen? Yeah, Jacqueline is nervous, but she's totally right. No ATP equals no detachment. If I don't have this ATP right here, this shiny thing to give to myosin, myosin is going to stay right here. Myosin is going to stay attached to actin, and it's never going to let go. As soon as myosin goes from being in its upright position to this bent over position here, and it's attached to actin, when we get here, no more movement happens. No more movement happens, but we're also stuck. Unless ATP comes along, myosin and actin are locked together forever, not moving. We said that in rigor mortis, your muscles get stiff when you die. Why do our muscles get stiff when we die? What's making them stiff? Any ideas?
So I think it would be easier to say it than to type it. It's a lot of,
Yeah, no, that's totally fine.
typing, I feel like. So they get stiff because they're stuck. Because
Exactly.
there's no ATP, so they can't move. Right?
Yeah. So what makes them stiff is myosin and actin are attached to each other. They're stuck attached to each other. There's no wiggle room back and forth. We can't detach from each other. Yeah, so Nicole mentioned the word too. We can't ever relax the muscle, because what would let us relax our muscle is when ATP allows us to detach them from each other. So rigor mortis, when your muscles get really stiff after you die, what's causing the muscles to get really stiff after you die is the fact that myosin is on to actin and it will not let go. So big picture, the stage of the cross-bridge cycle where I first start using ATP, where ATP plays the biggest initial role here, is getting myosin and actin to let go of each other. Otherwise, they're stuck attached together and our muscles are stiff. Now, the other thing I need ATP for, right, is to break it, to cock the myosin head. But we have to first detach myosin from actin before I can cock the myosin head. So in rigor mortis, here's my problem right here. I cannot detach myosin and actin from each other. That makes my muscles get really stiff. They're really stiff because they're stuck. There's no movement. I think I missed a question earlier, but someone had asked, I think, about how kind of this is a cycle that's kind of always occurring. It totally is. So we're always, as long as, the caveat being, as long as there's calcium floating around inside your muscle fiber, we're always going to go from step 1 to step 2, from step 2 to step 3, from step 3 to step 4. And if myosin gets back into the cocked position where it's standing upright and there's actin available for it to attach to, we're going to go back and we're going to do the circle of life. So as long as we always do each of these steps, this is going to happen all the time. When we want muscle contraction to stop, what we'll actually do is we will collect all of the calcium from our cell that was making this tropomyosin protein move, that was messing with the push pin, actually. When I don't want muscle contraction to happen, when I don't want to do the cross-bridge cycle, like I see here, I take away all the calcium, because the calcium is what allowed my push pin to let go of the spaghetti noodle, to allow the actin sites, those sites where myosin can attach, to be available. All these dots that we see here to be available. What questions do we have? Yes, there is a lot to remember. I think we had mentioned this either during our review or late last week, that this is this particular chapter is a process chapter. I know we hate process chapters, but a really great way to study them is to talk them out. So out loud, going through and trying to describe to somebody how the process of the cross-bridge cycle works. It's going to be really important that you be able to put it in your own words. So it's going to take time. It's going to take practice, but definitely try to do that. So let me see Nicole's question. When we move, so one of the videos embedded in the guided lesson shows you how this process works. As you move, so you're flexing your bicep, for example, as we're moving our hand up toward our shoulder, as you're doing that huge movement, moving your hand to your shoulder, this cycle is occurring super, super quickly. Like it's just going nuts. It's going nuts all over the place. And like I mentioned, there's a really great video in the guided lesson that shows you. We're looking at what happens with one myosin head attaching to actin. Remember when we were looking at those myosin proteins that there were heads all over the place along here. So literally you've got this crawling thing happening where right here's a myosin head attached to actin, and meanwhile over here there's another one, and there's another one over here. So the entire time that you are flexing your bicep muscle, your big bicep muscle, we've got a ton of myosin heads that are just going through this cycle really rapidly, completely. When we're done flexing our bicep, when we're trying to relax it back down, what will happen is we'll end up stuck in this phase. So as your muscle pulls back all of its calcium, that getting rid of that calcium here, let me go back to a slide that shows us calcium's job here. When I'm ready for a muscle to contract, what I'm going to do is I'm going to steal back all of this calcium that I had released. When I steal back that calcium, my push pin, my troponin, goes back to the right shape that allows it to hold my spaghetti noodle that I see right here, my tropomyosin, allows that to be held in place. Because then if I'm blocking the myosin binding sites, I can no longer do the first step of the cross-bridge cycle. I can't attach myosin to actin. So when I want muscle contraction to stop, the way that I do this is I block this step right here. If myosin and actin can't attach to each other, I can't do any of my other steps. But as long as I can do cross-bridge formation, and as long as I have ATP available, I will just keep doing this circle forever and ever and ever. We'll just keep going in circles here. So when you do a big body movement, it's not just one cycle, it's like millions of cycles. We go through this process super quickly, which is why we're burning through our ATP so quickly, because for us to do this once takes me ATP, but I'm going to do it, like I said, millions of times in the context of one body movement. So yeah, it keeps going over and over again. And there's, like I said, a really awesome video in the guided lesson about that. Let me look at Jacqueline's question. So when we talk about the role of ATP, there are two roles of ATP in muscle contraction. The first role of ATP in muscle contraction is allowing myosin and actin to detach from each other. That's the first role. ATP has to do that first to allow it to do its second job. Its second job is to cock the myosin head, to take myosin from being bent over to standing upright. That's the second job of ATP. So first job of ATP, detachment, making sure that myosin and actin aren't attached forever. Second job of ATP is to get the myosin head back into the cocked position where it's going to be able to attach to actin again. Does that answer your question? Yeah, so the putting it in the cocked position means putting it in the right shape. If you think about cocking a gun, where you're getting it ready to fire again, the cocked position just means the active position. Standing upright, ready to do muscle cross-bridge formation again, ready to do muscle contraction again. Okay, let me bounce back in the lesson momentarily to show you this. This is something that should look familiar from lab last week. I'm looking at something that goes from one Z disc to another Z disc. What is this thing in here called again? Do we remember what that structure is from one Z disc to another Z disc? Yeah, so Nicole's right. We are looking at on this slide here images of two different sarcomeres. I'll type that for our friends' video. Sarcomeres. Sarcomeres are the things that make up those myofibril tubes. So the reason that we saw them having that striped appearance, the striate appearance, is because of the sarcomeres. When we're talking about muscle contraction, the process of muscle contraction we can see in this sarcomere right here. Here's the way that we can tell, there are a few of the ways we can tell that this sarcomere up here is contracted. First way we can tell that you can see most clearly, if you look right here, see how I've got a myosin head that's attached to actin? Or if I look over here, I've got a myosin head that's attached to actin. multiple myosin heads attached to actin here. This sarcomere is in the process of contracting, and I can see that because myosin and actin are attached to each other. If you compare that to the sarcomere down here, notice how these myosin heads, none of them are touching the actin. This is because on these actin proteins, all these blue ones here, that spaghetti noodle, tropomyosin, must be covering the myosin binding sites. Myosin has nowhere to attach to on actin, so the process of muscle contraction isn't occurring. So first way I can tell that this is a contracted sarcomere is because myosin and actin are touching each other. They're pulling along each other. That's the first way I can tell. The second way I can tell is notice how much of my actin protein is overlapping with my myosin protein. See how there's just this little area here at the end where it's just actin? If I compare that down here, when we're looking at this sarcomere, there's lots of space where there's just actin on either side over here. In a relaxed sarcomere, myosin isn't grabbing actin. If myosin doesn't grab actin, actin likes to move this way. Naturally, my sarcomeres like to spread out as wide as they can. When myosin pulls on actin, myosin pulls actin toward the middle. It's going toward the M line. When the process of contraction stops, actin spreads back out and goes back out on its own. So when you think about you're doing that bicep curl that we talked about, when our muscles are getting shorter, what's actually happening is these little myosin heads grab at different places on the actin and pull it toward the middle. And they pull it a little bit more, and they just keep pulling it toward the middle for as long as myosin can attach to actin. As long as these two things can relate to each other, remember, as soon as myosin attaches to actin, myosin then is going to spit out that low energy ADP, and that's going to cause it to fold toward the middle. And when it folds toward the middle, it's going to move actin with it. It's going to pull actin toward the middle. And the same thing over here. When this myosin head spits out its ADP, that's going to pull actin toward the middle as well. So we're always pulling the actin proteins toward this M line, the midline of the protein. And when I do this with all of the sarcomeres in that big long tube, the entire tube goes from being really long to being a lot shorter, because everything's overlapping a lot more and squeezing together a lot more. Christina says it kind of reminds her of a slingshot. Yeah, we could think about it that way, how we're pulling and then we let go and relaxing. So yeah, we're going kind of back and forth with that. Gloria asked a good lab review question for us, and I'm going to have my friends in the chat help me out here. When we talk about the H zone and the I band, what was different between those things? Can anyone help us remember? Yeah, so Nicole's totally right for us here. She's saying that the H zone is where I find the thick filament in the sarcomere. The thick filament in our images here is the red one. So the places where I find the thick filament, that's right here. That's my H zone. Remember, H is a thick letter, so that's where I find the thick filament. The I band is the place where I only find the thin filament. So notice these places over here where there's just that blue thin filament. We've got this fun little spring protein. We're not really going to talk about that one, but we mainly just have actin here. So the actin proteins are in the thin filament. That's the I band. I is a thin letter. The H zone is in the middle where I just have the thin or the thick filament. And remember that we talked about how we also have this little section right here. This little section here is called the A band. The A band. The A band is where my thick filament and my thin filament overlap. So there's also this A band area too, where my H zone, the thick filament, overlaps with my I band, where the thin filament is, to make this area called the A band. So connection question here. I'm looking at my contracted sarcomere up here. Here's the three areas we just talked about. H zone, I band, and A band. When a sarcomere contracts, when it goes from looking like this down here to looking like this up here, what happens to the size of each of these things? H zone, I band, A band. Which of these things gets smaller? Which of these, the size of it gets smaller? So a couple of us are saying all. It's actually not quite all. Two of the three get smaller. H zone, I band, A band. Yeah, the first one that gets smaller is the H zone. Remember that the H zone is the place where I only have thick filaments. We'll type that up here. Only thick filaments. H zone. Remember down here, I had a big H zone right here, a space where there's only thick filaments. Hey, when I look at this sarcomere over here, do I even have an H zone anymore? Is there a space where there's just thick filaments in here? Y'all are like, she's tricky. I'm not tricky, I promise. There's no H zone left here. So the H zone, when a muscle is completely contracted, there might not even be an H zone. There might not even be a place where there's just thick filaments. Someone else mentioned in the chat too, the I band also gets smaller. Remember that the I band is the place where I only have thin filaments. So when I look over here at this contracted sarcomere, there is still an I band. The I band doesn't go all the way away, but it got a lot smaller. So the I band, where I only thin filaments, the I band also got smaller, the space where I only have thin filaments. At least it still exists. I still have a little bit of space for it, unlike the H zone where it's just gone. But the fact that the H zone is gone is because the size of the A band has gotten so much bigger. Remind me in the chat, what do I find in the A band? What do I find in the A band? Does anyone remember? Yeah, the A band has both. It has my zone of overlap. So the A band has thick and thin filaments. It has both of them. When I start contracting my sarcomere, the amount of space where the thick and the thin filaments are touching each other goes up. So my A band gets bigger when I do the process of contraction. My H zone and my I band each get smaller because the place where I only have one thing gets smaller when I do the process of contraction. I'm browsing the lesson here. I think I want to stop there for today because I don't want us to start something else and not have enough time for it. So help me out in the chat. What other questions or thoughts do we have right now? Okay, so the question that Gloria is posing for us here in the class is, which stage of the cross-bridge cycle is disrupted when a person dies? We've got one vote. Do we agree or disagree that it's stage 3? Yeah, looks like we agree. The stage of the cross-bridge cycle that is disrupted or completely stopped after death is stage 3 right here. Because stage 3 is when ATP comes in and allows actin and myosin to detach from each other. If actin and myosin can't detach, we can't go through and get myosin in the right position to be able either to connect to actin again and do the process of contraction, or to just stay detached from actin and allow actin to slide back out. So rigor mortis is a problem when they get stuck attached to each other and we can't detach them from one another. Detachment either lets me contract some more or it's going to let me relax. If I'm just stuck attached, I can't do either of those things. I'm in rigor mortis. I'm stiff and I'm dead. Yeah, it is weird, isn't it, Jacqueline? You wouldn't think that it's something as simple as splitting up the two proteins. You would think it would have something more to do with like powering movement, but the stage that needs that ATP, the stage that's most affected is the detachment stage, getting those two things to not attach to each other. Good. I'm glad that helps some. And we have some time to ruminate on this. There are some really good helpful videos in this guided lesson because this is a big process thing. So it's always good. The videos in the lesson will show you what happens in each of these stages. So definitely watch those videos multiple times to help you make sense of what's happening in each. Yeah, so Christina's asking about ATP hydrolysis. Gonna make me draw it again, right? Let's see if I can do it. Adenosine. And let's draw our phosphates. This is so much easier on a whiteboard. Making it work though. Technology just mostly hates us, not entirely hates us. Okay. So here is all my parts of ATP up here. I hold together, so ATP is adenosine triphosphate. Did I say T or D? I don't know. Sorry. This is ATP right here, triphosphate. So tri, I've got 3 phosphate groups attached right here. PO4 is attached. Remember that the energy of ATP is stored in the chemical bond right here between my second phosphate and my third phosphate. So if I go through and hydrolyze this bond, or if I chop up this bond, that's going to give me the energy that's stored right here and leave behind 2 phosphate groups attached to the adenosine. So when you look here and it says ADP, adenosine diphosphate, what I've done is I've stolen, let's take our little bond out there, I've stolen the bond that was between these 2 phosphates. Now I have an adenosine with 2 phosphates on it, that's ADP, 2 phosphates. And I've got this other phosphate that's just floating around, not doing anything. ADP, adenosine diphosphate, and this other phosphate group, there's no energy left right here because the energy that I have over here was stored inside this bond. When I chop up that energy, that changes the shape of my myosin head, and it gives me that energy as my myosin protein. But I leave behind this junk, ADP, and that phosphate. Does that help? Or is there another specific question that I can help with that? Nicole asked if there's a purpose for ADP. I mean, we are recycling it. So there's the purpose of I use it for recycling to build ATP again. I don't think there's anything in particular in your cell that like needs ADP to work. It really just is something that we're recycling to turn back into ATP. So the reason that it hangs on, so the reason it's attached here is just because I chopped it. So it's attached to myosin. And it's the same place here where ATP was. I chopped it up, so it just stayed attached because it's in a special place in that protein where there's space for it. But I physically squeeze it out of the attachment site when I bend over my protein here. So there's not a purpose for it except for just, it doesn't bother me that it's there. So it hangs on until I spit it out and change my shape. It's kind of like if you have a cutting board, for example, and you're chopping an onion, until you put that onion into the pan that you're cooking in, the onion is going to be there on the pan. You've got the cutting board. It's just kind of this is the place where I cut my ATP is here on my cutting board here. It's going to stay on my cutting board until I spit it out and don't have it anymore. Any other last minute thoughts? Got some mind blown going on. Anybody? Yeah, so good questions about next week. Next week, we are continuing to work on lesson number 9. The lesson number 9 homework assignment is due next Friday. So keep in mind, it's due next Friday, not next Sunday, but next Friday. We're going to take like a week and a half of class time on it. For the live sessions next week, we are going to do live sessions. I'm going to do Monday morning, Monday night, and Tuesday. You guys are my friends who come to the sessions regularly. Would Tuesday at 9:30 be better or worse than Tuesday at 11? Options are Tuesday at 9:30 or Tuesday at 11. Earlier's better? Earlier's better for me too, I think. Okay, so Nicole says 11. So I think I'm probably gonna do I know we're kind of tied, right? I think I'm gonna do the 9:30. I know not everyone will be able to make it, but what we will do Monday morning at 9:30. I will either do lecture or lab. Monday night I'll do the opposite, whatever it is, and then we'll use Tuesday at 9:30 to wrap up the lecture lesson. I'm gonna have office hours. Probably it's either going to be Wednesday or Thursday. I'm going to be traveling, so I can't vouch for my internet. We'll see how it does. But we will do our live sessions on Monday. So here, let's type our schedule. Monday at 9:30, Monday at 5, and then Tuesday at 9:30 will be our sessions, our scheduled sessions next week. Thinking about, I bet we will do lab Monday morning, and then we'll do lecture Monday night and Tuesday morning to make sure we have what we need for that. So for lecture, Nicole, I'd say go ahead and try to work through the rest of it. I bet that we'll spend most of our lecture time the first time talking about what's called excitation, contraction, coupling, which is, okay, you've decided you want your muscle to contract. How do we make muscle contraction happen from a neuron side of things? I would bet we'll spend a lot of our time doing that. You get to study about Botox and about sarin gas, so some fun correlations to make. I'm looking at the thing now. I'd say try to, for Monday's session, try to get all the way through the top part of page 13. So the 3 types of muscle fibers, I bet, will be what we talk about Tuesday morning. So try to get everything finished up through sarin and Botox. to get you ready for Monday night's session. And then we will, if we have time, we'll talk about the 3 types of muscle fibers Monday night. Otherwise, we'll plan to do that on Tuesday morning and just kind of review any last minute questions that we have about lesson number 9. Christina has a question. What's that question, Christina?
Is there a new group wiki posted yet?
Not yet. It is on, I literally have a to-do list. It is on my to-do list. So hopefully tonight or tomorrow, I will get that next group wiki up for you. What I'll say about this one, because unit number 3, the one we're working on now, is really process driven, you may not be able to answer many of the questions right now since we just started, especially because we're going to talk neuron stuff too. The brain stuff will be, so here's to give you guys a little bit of hope. Lesson number 9 and lesson number 10 are really process heavy. So today we talked about the process of the cross-bridge cycle. Monday we'll talk about the process of excitation, contraction, coupling. Lesson number 11 is not a process. Lesson number 11 is all the parts of the brain, learning what the parts of the brain do. So if you more prefer the straight memorizing stuff, it's going to be a little bit easier to straight memorize the stuff from lesson number 11. So a little bit of relief coming in number 11. We just got to get there first, right? Number 9 and number 10. All right. Well, I think I'm going to go ahead and wrap up the recording. I'll stick around for a couple minutes with any last minute questions you guys have. Otherwise, check that schedule. I'll try to get it posted at the latest Saturday, the schedule of what we're doing next week. But yeah, I think we'll do Monday morning lab, Monday night lecture, and Tuesday morning lecture as well. So check for that schedule, check for those recordings, and we will work our way through this lesson together.